Hello, my name is Yusin Laston. Topic: Targeting Antithrombin to Treat Hemophilia. Definition: Hemophilia is a tension bleeding disorder that occurs from a deficiency of coagulation factor 8 or 9, which causes hemophilia A or B respectively. Any characteristic big spontaneous and traumatic bleeding as the motion, showing in the body cavity.
So, what is hemophilia? Hemophilia is a bleeding disorder, a problem with a person's blood that makes them bleed longer than a person without hemophilia. No one can catch hemophilia. It's an inherited condition you have from birth, for your whole life. It's just part of who you are, like the color of your hair or the shape of your nose. Here's how it works. Your blood travels through your body in tubes called arteries, veins, and capillaries. Normally, when there's a tear or hole in one of these, bleeding starts, and different parts of the blood go to work to plug the hole. Blood cells called platelets gather at the opening, and other blood parts called clotting factors help hold the platelets together to form a clot that stops the bleeding. When a person has hemophilia, he doesn't have enough of one of those clotting factors he needs to form a clot, and he keeps bleeding longer than he should. Each person with hemophilia is missing one of two kinds of factors. If you're missing factor 8, you have hemophilia A. If you're missing factor 9, you have hemophilia B. Either one can be mild, moderate, or severe. Without regular treatment, having hemophilia means you'll bleed longer than you should if you get cut. But you don't have to have a cut to have a bleed. Sometimes, especially when you're active, the tiny capillaries inside a person's body will bleed, causing a bruise or pain in the joints. Not dealing with those kinds of bleeds can cause a lot of problems later, including arthritis at a young age. But the good news is, there are good treatments available that can help you manage hemophilia effectively. Treatments that replace the missing factor help your blood clot more like a person who doesn't have hemophilia. These treatments, when used properly, help you keep your bleeding under control so you can live a long, healthy life and follow your dreams without hemophilia ever holding you back. To learn more about hemophilia, visit worldhemophiliaday.org.
Application in nursing. The treatment of hemophilia is relevant symptoms. It simply consists of the contribution of the action or deficiency coagulation factor in the patient. To do this, complete transfusion or intravenous injection are carried out, and as well as frozen plasma or concentration or plasmatic factor and concentrated of recombination factor.
Hemophilia A is an inherited bleeding disorder caused by a deficiency in factor 8 activity. The current standard of care includes prophylactic intravenous factor 8 infusions. However, these infusions may result in the development of anti-factor 8 antibodies that can inhibit efficacy and increase morbidity. Intravenous bypassing agents such as activated factor 7 and activated prothrombin complex may be required to counteract these effects. Emicizumab is a recombinant humanized bispecific monoclonal antibody that binds to activated factor 9 and factor 10, brings them into proximity, and restores the scaffolding function of the missing activated factor 8. In the Phase 3 International Open-Label HAVEN study, investigators randomized in a 2-to-1 ratio 53 participants with hemophilia A of any severity who had documented high factor 8 inhibitor levels and episodic treatment with bypassing agents to receive weekly subcutaneous emicizumab prophylaxis or no prophylaxis. An additional 56 participants who had a history of prophylactic use of bypassing agents or who were not able to enroll in other study groups were assigned to treatment with emicizumab. The primary endpoint was the reduction in annualized bleeding rates after 24 weeks of treatment. Emicizumab prophylaxis resulted in a statistically significant decrease in the annualized bleeding rate. to 2.9 as compared with 23.3 in the no prophylaxis group, which represented a decrease of 87%. In addition, 63% of the participants randomized to emicizumab prophylaxis had no bleeding. Thrombotic microangiopathy and thrombosis were serious adverse events reported in 4 participants receiving both emicizumab and activated prothrombin complex concentrate, but these effects resolved after cessation of the bypassing agent. The authors conclude that emicizumab prophylaxis prevented or substantially reduced episodes of bleeding in patients with hemophilia A with inhibitors. Full trial results are available at nejm.org.
4. The output of inhibiting antithrombin is that it removes that natural thrombin inhibitor, factor 2A, and the next hemostatic effect is less severe bleeding, which has been demonstrated in most models for hemophilia A, improving hemostatic after antithrombin display. Cons. Treatment is very expensive, costing and thousands of dollars and care for the advanced adult, and its complications often aggravated the disease, impair quality of life, and threaten orthopedic and surgical regimen and social life.
Hemophilia is a genetically transmitted disease which largely affects males. Females are the carrier. In the United States, there's about 20,000 people with either hemophilia A or hemophilia B. Around the world, there's about 400,000 people affected by hemophilia. It's about 1 in 5,000 live births of males. There are other bleeding disorders which similarly afflict both males and females. The most dominant one is called von Willebrand's disease. And the community has experienced a wealth of investment in research and development and new products that are continually improving the lives of people with hemophilia. Just over the last 5 years, we've had extended half-life Factor 8 and Factor 9 products introduced into the marketplace. Just last year, a disruptive therapy from Genentech called acuzumab, which bypasses the coagulation cascade, has been brought to market, which promises even better results for many patients with hemophilia. And probably within the next 12 to 36 months, there'll be multiple companies bringing gene therapy products to market. So it's really remarkable that over the 42 years I've been in this space, we've gone from cryoprecipitate to factor concentrates to highly purified extended half-life concentrates to soon gene therapy, which is almost a cure for hemophilia. As I mentioned earlier, there's only about 20,000 people in the United States with hemophilia. So you have to wonder why this level of investment. Well, there's a couple of reasons for that. One, there's a great deal of interest at the pharmaceutical level in the technology around bringing a gene therapy product to market. So hemophilia, because of the size of the Factor 9 molecule, was an obvious first target for testing the viability of gene therapy, not just in hemophilia, but in a wide range of diseases. As you heard earlier today at the Rare Disease Conference, there's something like 600 different compounds in various stages of clinical trial for a wide variety of diseases. All of that started in the hemophilia space, going back 25 years ago. But I think the other reason for the investment is a very active patient community who have insisted on investment in hemophilia. And we were one of the original rare diseases, if you will. There was actually a way to treat hemophilia. The patient community was organized. The manufacturers in this space were committed to making lives better for people with hemophilia. And the result of all of that investment is really paying off, not just for people with hemophilia, but people with different chronic and rare diseases around the world.
The Ministry of Public Health has 16 hospitals where its foreign compensated for to the hemophilia patient, thanks to the work of a multidisciplinary team of health professionals, internists, nurses, dentists, traumatology, rehabilitators, pharmacists, and social work. Is correction cards or patients of which has hemophilia A, A4 has hemophilia B, A9 has bone wheelbarrow, and 5 with factor hearing deficiency.
Hemophilia is a rare genetic disorder that prevents blood from clotting properly. As a result, prolonged external bleeding can occur spontaneously or due to injury, bruises, dental work, or surgery. Internal bleeding can also be incredibly dangerous for people with hemophilia. Deep bleeding inside joints and muscles can lead to permanent disability or may be life-threatening, especially when damage occurs to organs. Hemophilia occurs when the genes that are vital to creating clotting factors are defective. Clotting factors are proteins in the blood that help our bodies to stop bleeds. There are 12 defined clotting factors. Hemophilia A is caused by a mutation in the gene that creates a deficient amount of clotting factor 8, while hemophilia B is caused by a deficiency in clotting factor 9. Different deficiencies, each caused by a different genetic defect. The most common existing hemophilia treatments administer clotting factor replacement therapy into the bloodstream through an infusion. These treatments are required throughout a person's entire lifetime to prevent or treat bleeding. However, gene therapy could offer a one-time treatment. With gene therapy, a functional version of the missing or defective gene can be delivered into a cell to produce the missing factor. Functional genes need to be delivered into the cells using a vector, which is often derived from viruses. Don't worry, all viral genes are removed, and the vector is engineered to only deliver therapeutic genes. To treat hemophilia A and B, the vector would be targeted to a person's liver cells via infusion. To explain more about the liver's role, here's Dr. Denise Sabatino.
The liver is an important organ that is responsible for creating a variety of elements in our blood, including clotting factors 8 and 9. In individuals with hemophilia, the genes that produce these factors are defective or absent. By introducing a vector containing the functional gene to liver cells, the liver can produce and secrete functionally active clotting factors back into the blood to prevent bleeds.
Since the liver grows with us from childhood to adulthood, gene therapy trials are currently being conducted in eligible adults with hemophilia. This helps to avoid the therapy's beneficial effects from becoming weaker or wearing off as a child and their liver grows with age. Researchers continue to optimistically monitor gene therapies in clinical trials until they can safely and effectively prevent bleeds by making up for the deficiency. But if a bleed does occur during or after receiving a gene therapy, it is important to know that the use of any standard of care treatment can still be used. An individual with hemophilia should carefully discuss gene therapy options with their hematologist or hemophilia treatment center. For more information and resources about gene therapy for hemophilia, visit asgct.org.
In conclusion, the potential to decrease bleeding in hemophilia with or without inhibition with a single weekly subcutaneous dose would be life-changing. It will be a question advance To improve protection from bleeding with a simple subcutaneous injection or with in a less injection one.
Normally, after a cut and damage to the endothelium, or the inner lining of blood vessel walls, there's an immediate vasoconstriction or narrowing of the blood vessel, which limits the amount of blood flow. After that, some platelets adhere to the damaged vessel wall and become activated, and then recruit additional platelets to form a plug. The formation of the platelet plug is called primary hemostasis. After that, the coagulation cascade is activated. First off, in the blood, there's a set of clotting factors, most of which are proteins synthesized by the liver. And usually these are inactive and just floating around in the blood. The coagulation cascade starts when one of these proteins gets proteolytically cleaved. This active protein then proteolytically cleaves and activates the next clotting factor, and so on.
Sign up for a free trial today and watch the full video on osmosis.org.